Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay on the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on our screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Bernardo Cordovez from Halo Labs.
Hello everyone, my name is Bernardo Cordovez and I am founder and chief science officer of Halo Labs. Today I am very excited to tell you how the Aura PTX can help you characterize protein aggregates and extrinsic particles from cell line development through product release. And some of you may not know this, but actually particles are the most common cause for product recalls. And the reason for that is that particles, and particularly subvisible particles, which are micrometer scale particles, are a critical quality attribute. They present a safety concern. They can be immunogenic and even cause capillary occlusion. They result in reduced drug efficacy. And the presence of particles in a biologic formulation essentially indicate that a product may have a short shelf life. And as a result of all of these, the FDA tightly regulates the amount of subvisible particles allowed in a product, following USP 788 guidelines focusing on particles larger than 10 microns in size. Biologics are plagued with subvisible particles, and they can be intrinsic, so essentially the drug product itself, these large molecules, can aggregate, indicating to product stability issues. And in the image on the left, we see large subvisible protein aggregates, while on the image on the right, we see subvisible aggregates of a gene therapy. And these are common, and they can be big, and there can be many of these. But particles don't always come from stability issues. They may actually arise from external environments. And in this case, we can see an image that distinguishes between large protein aggregates and plastic particles in a given formulation. Fundamentally, the Aura product line is about measuring biologic aggregates and product impurities, and it does so by imaging, sizing, counting, and identifying every single particle in a given formulation or sample. And the output of this characterization is to ensure that you have a product of the highest quality, stability, and purity across all stages in the bioprocess. The Aura builds off of the USP 788 compendial method to enable high throughput, high sensitivity, subvisible particle analysis and ID. And in the next few slides, I will show you how it works and how it can help you. The process for imaging, counting, and sizing particles in the Aura system is called background and membrane imaging. And it is very simple and it only takes 3 steps. We have a proprietary 96 well filter plate, and the first step is to background the wells of choice, to then filter the samples of interest that we're measuring, and then to reinsert that plate back into the instrument and re-imaging. It's that easy. Let me show you the results that you get. At the first level, you get images for every single particle present in every single well. And each of these particles is analyzed independently, resulting in key morphologically relevant as well as size information. This particular particle measures 57 microns in diameter, its largest extension is 77 microns, and it's not circular. At the next level, we have sample-wide information. That means characterizing how many small particles and how many large particles are present in a given well. And this graph on the right here is a standard particle size distribution for a protein biologic where there are lots of small particles, just a few microns in size, and very few large particles. And that's important because we do not want to have many big particles. But one step up, this is a high throughput instrument. So we can quickly characterize the entire particle population across 96 wells very quickly. and obtain experiment-wide results. So we can see how many particles for a given sample are bigger than 10 microns, bigger than 25 microns, and do traditional USP type passing analysis as well. And all of this is very powerful and interesting. However, can we really say what these particles are? And this is a key element to USP 788 because it's not just about passing. It's not just about having low particle counts. It's about understanding what is present in our samples. And the short answer with Aura is yes. We use proprietary fluorescence membrane microscopy to ID every single particle in a sample. So once particles have been imaged, sized, and counted in background and membrane imaging, we then add a fluorescence stain of interest to a particular well, and then see which of those particles fluoresce strongly above a background, and those are fluorescence positive particles. It's a concept pretty similar to those of you who've worked with flow cytometry. This is imaging solid phase cytometry. And every single one of the dots on this graph on the right represents a given particle, which after being stained will probe for specific chemical and biological characteristics. So we just discussed about imaging, counting, sizing, and identifying particles. And I ask you, so at what point should one be interested in actually measuring particles? Is it at cell line development, perhaps developability? You've already have a couple of candidates that you want to push through and see if they're manufacturable. Is it in traditional formulation development or during release or even later? Well, most traditional technologies, the legacy techniques, focus on measuring after there's already sufficient sample that has been able to be produced in the pipeline. So legacy instruments work in the middle to late formulation, and they don't really work all that well. And one thing that tends to happen here is that people forget why USP 788 exists in the first place, and that is to mitigate particle formation and particle presence no matter the source. So the right answer of when should you measure particles? It should be in the entirety of the process because particles are a critical quality attribute. The bigger they are, the more important and more dangerous they can be. And therefore you should know from the very beginning that your product is void of particles and that it is stable and that you do not have contamination issues. And the Aura allows you to do just that. The ability to measure product quality, purity, and stability is related to particles from the beginning to the end of the process. For protein therapeutics, we specifically have the Aura PTX, and it's designed again for measuring stability and purity from cell line development to product release of these very important biologics. Let me show you how. And the BioProcess will start with cell line development. And in particularly, CHO cell lines have been the ones that have been the most vetted and the most common for producing monoclonal antibodies. And right now in cell line development, the selection process is to pick those who produce the most antibody, the highest titer. However, producing the most antibodies doesn't necessarily correlate with producing the most stable antibodies. As a matter of fact, you may be choosing winners and pushing downstreams molecules that are inherently unstable. And with the Aura PTX, using specific stains that probe for the presence of aggregates of your product of interest, we can quickly determine if a cell line is stable or unstable. On the left, the green fluorescence means that those are protein aggregates being produced by the CHO cell lines, while the red on the right, those are just individual cells, CHO cells, that produce monomers, but they did not produce protein aggregates. The output is not just images, but because this is a high throughput system, you could then rank cell lines according to their stability and figure out on a pre-screen basis which cell lines you should push forward. The next step is developability assessment. So you have some cell lines of interest and some molecules that you want to push through to see which of those are going to be manufacturable by subjecting them to a battery of excipients and different buffer conditions. In this case, we have 3 different proteins subjected to an array of standard buffer conditions. And we can see that protein 2 produced the most counts under all sorts of different buffers, protein 1 produced the least, while protein 3 produced more than protein 1, indicating that protein 1 is probably the candidate that you want to push through. And this is possible because typically tools right now are unable to handle low sample volumes, but the Aura can. As a matter of fact, we see measurement linearity from ultra low volumes as low as 5 microliters, as low as 10 microliters as well, with high repeatability. And this is unique because competitive technologies can only process hundreds of microliters for a single replicate. The next step is biologic formulation development. And here, you really want to be able to measure polydispersed samples such as protein aggregates or other biologic samples with high precision. In this image, we're showing how we have low CVs measuring human IgG aggregates caused by rotation stress, as well as protein plastic mimics made by the National Institute of Standards and Technology. And if you add the amount of plastic to proteins, the total sum adds up. But in addition to this, the Aura system has the ability to distinguish protein particles from other particles. The proteins aggregates as well, compared to these protein plastic mimics, they look the same under standard microscopic techniques, but using fluorescence membrane microscopy, we can easily distinguish the two and tell you which percentage of the population is proteinaceous versus none. And we do this in high throughput for measuring every single particle across the entirety of the sample. As a USP 788 compliant method, we can show that we can validate sizing, measuring the appropriate beads correctly and within measurement error, and for the first time also enable precise counting and membrane microscopy systems, here using standard 33,000 particles per mil count calibration standards, showing that it's repeatable across units and across users. So the system can be validatable, and it is available with 21 CFR compliant software. Towards the tail end of the process, there are post-marketing reasons why you may want to do particle analysis. One common one is degraded polysorbate. So the very excipients and surfactants that are used to stabilize samples can degrade over time due to oxidation and other types of stresses, and they may look identical to protein aggregates. We have a high throughput system and ability to distinguish proteinaceous particles from degraded polysorbate fatty acid particles, as shown via imaging, and this image on the right, where the population separation is complete, and you can obtain deterministic differentiation between the two populations. So why Aura PTX? Honestly, why not Aura PTX? It's protein stability from cell line development through product release, measuring 100% of your particles and knowing what every single one of those are. It's compatible with USP 788. It's fast. It takes a few hours to measure hundreds of samples as opposed to months. And it comes with 21 CFR compatible software should you choose to take this into manufacturing. And it's a very easy system to use. Thank you so much for your time, and I look forward to your questions now. And after the Q&A, if you have any questions, please contact us at sales@halolabs.com. Thanks again.
Okay, great. So the first question is, how does this compare to the MFI or FlowCam?
Hello, everyone. That's a good question, and we get it often. The BMI technology is more sensitive than flow imaging methods. As I explained, we have a higher refractive index contrast, meaning that we can see particles that would be invisible for flow methods, including MFI and FlowCam, and therefore do a better job. In addition, this is a compendium. So what that means is that there's actually counts established for membrane microscopy, while there aren't any for flow methods.
And how does this compare to LO?
It's a good question. And on the same vein, the light obscuration is also a liquid-based imaging, not imaging detection method, and therefore it's very well now diagnosed in the literature that it tends to undercount particles significantly, and that it's only good at measuring particles essentially that are hardened with a big refractive index contrast with that of water. So essentially what this means is that it also undercounts particles significantly, and it's certainly, for lack of a better word, a danger, particularly if you're using it upstream when making these determinations is essential.
Is the sample analyzed as a wet droplet or filtered dry?
It is filter dried following the USP 788 protocol, and the main advantage is that when you do that, you'll have a high refractive index contrast, meaning particles that would be invisible for flow methods will be revealed with this BMI technology.
Can you describe the source or how you derive secondary antibodies specific for aggregated IgG?
Good question. We more than specific for the aggregated, it was specific to that product. So it was just a monomer binding a secondary. But essentially, we basically used the customer's established ELISA technique. So they had like the sandwich as a primary and secondary. Everything was already established. We just basically used their methods, and that's what enabled us to basically prove that in fact it was their product that was aggregating as opposed to any other protein aggregating.
Is it possible to distinguish different proteins in a sample?
Yeah, and similar to the previous question, the answer is yes. So for example, we have the ability, there's like a generic protein stain. If you wanted to look at, for example, thioflavin T is really good at just looking at generic protein aggregates, but then previous to the question before, you can use antibody labeling that are specific to proteins of interest. You can different primaries, secondaries, and reporters that are specific to the proteins that you're trying to interrogate to see what are the proteins that are aggregating. So that's a really good question.
And what is the pore size of your membrane then?
400 nanometers. So the pore size are 400 nanometers, but we're basically only detecting particles larger than a micron. So everything that is smaller than 400 nanometers, like all your monomers, dimers, and even nanometer scale particles will flow through. Basically, everything that's larger than 4, particularly biologics, they're sticky, right? They'll remain behind, and then our detection for this tool is about 1 micron in size. These are really good questions. Thank you, everybody.
Yeah, if you have any more questions, feel free to type them in now, and we will pass them on to Bernardo.
Yeah,
follow up with you directly.
Absolutely. Feel free to reach out. I really appreciate the questions.
Okay, well, I think that is all the questions we have at the current time. So thank you, Bernardo.
Thank you. Appreciate it. Thanks, everybody, for attending.
The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thanks, everyone.